Hello, everybody, and welcome to our Hoard Dairyman monthly webinar. My name is Abby Bauer, and I will serve as the host for today's program, which focuses on dairy cattle reproduction and more specifically, embryonic losses. This webinar will explore the important periods of embryonic loss following insemination in dairy cows and what we know about the causes and solutions to the problem. If you're listening to the presentation live, you have access to a copy of the slides. You can find that in the GoToWebinar control panel in the handout section. Just click on that link down in the handout section and you'll find a PDF of the slides down there. If you have any questions for our speaker, you can type those into the questions section of that control panel and we'll address those after the presentation. Today, it is my honor to welcome our speaker, Dr. Matthew Lucy. Dr. Lucy is a professor of animal science at the University of Missouri in Columbia. He received a Bachelor of Science degree from Cornell University, a Master of Science degree from Kansas State University, and a PhD from the University of Florida. He was a postdoc at Monsanto in St. Louis before accepting a faculty position at the University of Missouri in 1994. Dr. Lucy's current research program examines the metabolic processes regulating the endocrinology and fertility of dairy cows and explores practical methods that evolve from this research. He has earned many recognitions over his great career, including the ADSA Pfizer Animal Health Physiology Award and the ASAS Animal Physiology and Endocrinology Award. He served as editor-in-chief for the Journal of Dairy Science and was the first editor-in-chief of the Journal of Dairy Science communications publication. He became a fellow of ADSA in 2019 and received their Award of Honor in 2020. He currently serves as the host of the ADSA podcast, Dairy Digression, so you can hear him there if you want to learn more from him. So Dr. Lucy, welcome to the Hoard Dairyman webinar, and I look forward to your presentation, which is titled, Understanding and Preventing Embryonic Losses in Dairy Cattle.
Thank you very much, Abby. It's certainly a pleasure to be there, and I'd like to compliment you on your magazine. I really enjoy reading Hoard's Dairyman, and I enjoy seeing all the speakers you have for your webinar series. And today we're going to talk about embryonic loss in dairy cows and cover some of the more recent developments in this topic. Before I begin, or as I begin, I just want to recognize a few people. You'll see some of their data and some of their comments here. I just want to recognize them because a lot of what I'm going to talk about has to do with some of their own contributions to the scientific literature. The first is Pete Hansen from the University of Florida and his work on embryology. And of course, I think we're all familiar with Milo Wilbank and Paul Fricke from the University of Wisconsin. So I just want to give a little bit of shout out and recognize them for their contributions. I'd also like to recognize my good friend and collaborator, Dr. Scott Pope from the University of Missouri. Anything I do or present in this webinar always has Scott's involved. We work together on all these projects, and he's a really good friend, and I always like to tell people we want the best people on the team, regardless of how old they are. And I only say that because I'm actually a little bit older than Scott, but I just want to recognize him because he's such a good friend and such an important part of our research program here at the University of Missouri. I'd also like to recognize either both past members of the laboratory. This is 4 Brazilians that previously worked with me, as well as the current members of my laboratory. And all these individuals are so important to contributing to the research program that we have here at the University of Missouri. I want to just remind everybody that we're going to talk about embryonic loss, and I'm not going to give you any. I'm going to give you a lot of information. I'm not sure I'm going to give you any magic solutions to prevent embryonic loss. But let's just remember that what we want in terms of preventing embryonic loss is to start with a clean, comfortable, healthy cow and good body condition. And of course, that all comes down to good management. We of course also want good genetics because I use the term good cows. Good cows make a difference. And modern genetic selection indices are designed to increase the health, fertility, longevity, efficiency, and productivity of dairy cows. And you just can't, replace the really high quality cow in preventing problems like early embryonic loss. And the other thing you we need to pay attention to, because a lot of what I'm going to talk about today will have to do with just uterine health and uterine function. And in order to decrease embryonic loss, we really need to do everything possible to reduce the incidence of postpartum diseases, not only metabolic diseases, but also reproductive diseases, and particularly metritis and endometritis after calving. And I think most people believe that there's lots of causes of uterine disease, but it all kind of starts with the calving pen, and a clean environment in the calving pen is so important to maintain a clean and healthy uterus. So when we think about getting cows pregnant, I think about it as 3 steps. And we want to start with a healthy uterus. That includes that's as I mentioned, a healthy uterus that's at no uterine disease. We want a healthy ovary. We want the cow to be cycling, and we got to put semen in the cow. And I think that's what we've learned over my career, is that it's so important to get semen into the cow. And we're going to talk about that towards the end of this podcast. And of course, steps 1 and 2 are heavily dependent on management, as well as the endocrine and metabolic state that supports milk production. So a lot of the things we know that cows early postpartum are under a considerable amount of stress associated with initiation, lactation, and production of milk. But we need to minimize that stress, and in particular, we want to make sure that the strain that the cows feel in response to that stress is the least. And everything depends on good people and good management, except step 3, including step 3, unless of course the bull is doing it. And of course, we know on most dairies we don't have too many bulls putting semen into cows. Now, I think when we think about dairy cow fertility, it is kind of amazing. I mean, I don't know, we sort of take it for granted that, we can get cows pregnant and have calves, but it is quite amazing, because in order to produce a calf, there's a whole series of things that have to happen, and they have to happen in sequence, and they cannot fail. And there's lots of challenges along the way. And, the first challenge we have is anestrus, or anovulation, and we want to make sure cows are cycling. And we deal with problems with circulating hormone concentrations. And I think in the dairy cow, there's a lot of discussion about estradiol and progesterone, and challenges in terms of making sure we have estradiol and progesterone in the postpartum cow. And then we deal with poor quality gametes and embryos, which we'll talk about in a second. Issues with uterine health, which we'll talk a lot about today. But all these things, all these challenges that postpartum cows receive, all these things contribute to embryonic loss. So embryonic loss is sort of the summation of a lot of different challenges that face that high-producing cow during the postpartum period. So I don't want to talk, just before we talk about embryonic loss, well, I'm going to talk about embryonic loss, but I want to just talk about the things that have to happen to produce a calf. And of course, the first one is fertilization. Sperm must reach egg. And we think that the efficiency of fertilization is generally pretty good in postpartum cows. We think about, if we're using a good sire and good semen, we'll get about 85% of those animals with a fertilized egg after breeding. The next thing that has to happen is that fertilized egg must go through a series of cellular divisions, and we call these cleavages. And during this period of cellular division, they turn on their genome. In other words, they turn on their genes so they can basically start development. And as the embryo develops, it divides, and it does something called compaction, and that's where the cells kind of come together. That's about day 5 or 6 after breeding, and that's when the embryo actually leaves the oviduct and enters into the uterus itself. But it's not done that. It has to go through this process of cavitation, and during cavitation and the formation of the blastocyst, it's actually forming a placenta, the earliest placental parts, and forming the earliest parts of the embryo, or the intracell mass. And then finally, it must hatch. Although we think about chicken eggs hatching, bovine embryos also must hatch and exit the zona pellucida, and that's about a week and a half after first breeding. Now, After hatching, the embryo continues to grow and develop. And this is a really critical period in the embryo's life because basically during this growth and development period after hatching, it forms two different structures. It initially becomes spherical, maybe 10 days after breeding, and then becomes what we call tubular. And tubular is a critical point for this critical place for this embryo to get, because it's at this point it's going to signal the mother that it's there. And so during these early phases, the mother doesn't really know she's pregnant, but eventually once you get to this tubular phase of embryonic development, that's when the embryo is going to say, hey, mom, I'm here, don't kick me out, let me essentially live. And we talk a lot about progesterone, and progesterone is a really important hormone from the corpus luteum. And the reason we're so focused on the progesterone is progesterone maintains pregnancy, it stimulates the uterus, and it's also the hormone that sort of stimulates the process that this embryo gets to this tubular form of development so that it can signal the mother and tell the mother that it is present. Now, why is it so critical? Well, during the estrous cycle, the uterus is actually not set up biologically to maintain a pregnancy. It actually is set up biologically to cycle, to go through repeated estrous cycles. And the only time those estrous cycles are interrupted is when there's an embryo, present inside the uterus. And so that embryo has to achieve sufficient size so it can produce another molecule called interferon tau, and then interferon tau blocks prostaglandin release so that the corpus luteum can be maintained during pregnancy. So this is an important milestone during early embryonic development, and one of the things that we look at, because that embryo has to develop fast enough to prevent the corpus luteum from regressing so that it can survive. Now, following the filamentous embryo, there's a series, the conceptus continues to grow and the placental forms, and you can see these different pictures of a developing bovine pregnancy. And so the little thing in the middle here, that's actually the fetus. But you can see the rest of this is all placenta. And one of the things I want you to notice right away here is that these 28 and 30 days placentas, there are no cotyledons formed in these placentas yet. In other words, this is a period before which the placenta hasn't really firmly attached to the uterus. And this is important. Now, many times we think about this is the period we see all the embryonic loss between 28 and 60 days, and that's true. And one of the reasons we see a lot of embryonic loss during this period is that the placenta hasn't really attached fully to the uterus. It's not till the later stages of pregnancy, maybe 45 days, when we finally see these placental attachments to the uterus begin to form. And by the time we get out to 55 or 75 days, for example, they are fully formed. So one of the reasons we talk a lot about embryonic loss, for example, before day 60 of pregnancy, is because the placenta is not fully formed before day 60 of pregnancy. After day 60 of pregnancy, the placenta is fully formed, and we really don't see that much embryonic loss at that point. Now, one of the things that I want to bring up right away is the fact that there is a tremendous amount of diversity in terms of the development of the pregnancy. And what I mean by that is different pregnancies develop at different rates. And this is something that many different individuals are studying. I know that Milo's been studying this and Richard Pursley's been studying this, trying to understand why different pregnancies develop at different rates, and then what are the implications of that. So as an example, this is a study we did a long time ago, but we pulled both of these fetuses out of a cow that was 35 days, out of cows that were 35 days pregnant. And you can see the tremendous difference in size between those two fetuses. You can also see the tremendous difference in the circulating concentrations of pregnancy-associated glycoprotein, which is an indicator of placental development. And so that's one thing we're trying to understand. Like in 2 cows, why do we get different rates of pregnancy development? Now, in this specific experiment, we were looking at lactating and non-lactating cows and studying that. But even in a group of lactating cows, you're going to see quite a diversity in terms of how fast that pregnancy develops. So when does embryonic loss occur? And this is something I think that we're starting to understand a lot better. You know, Dr. Wiltbank in his excellent review published in 2016, and I've got the reference there, he talked about different periods. The first week of pregnancy, he talked about embryonic loss between 8 and 27 days of pregnancy when that embryo is elongating and undergoing maternal recognition of pregnancy. Talked about embryonic loss during placentation. That was the period I just spoke about. And then he talked about embryonic loss later. And I mentioned that this later embryonic loss, 60 to 90 days, is quite low in cattle because the placenta is fully formed at that point. And I'm going to just do a quick amendment to Dr. Wiltbank's list here. I'm just going to add I'm going to break down this 8 to 27 day period that he had. I'm going to break that down into 2 periods because I think they're kind of different in terms of what's going on. So I'm going to break this down into 2 periods, a period of pregnancy development between 8 and 18 and a second period of pregnancy development between 18 and 27 days of pregnancy. So the reason scientifically, I think it's important for members of the dairy community to understand that pregnancy loss is really hard to study because a lot of times we just don't know what we've inseminated the cow, but we really don't know whether or not she's pregnant. And so what do I mean by that? Well, if you think about breeding a cow and then, you know, during the first week after breeding, we don't have any easy ways to know whether or not we've got a viable embryo inside that cow. And the only way we can understand that or study that is to actually attempt to flush the embryo out of the cow and look for it under a microscope. And you can do this on a small scale, but to really understand embryonic loss, for example, you need to do it on a large scale in hundreds or maybe thousands of cows. And then that's just really impractical. So part of the reason there's so much confusion is because we just don't have a lot of good information, particularly during this early periods. Now, during later periods, this period from 8 to 18, we can tell whether or not a cow is pregnant using an ISG15 test, which is a test that's based on interferon tau production. And after that, we can use a PAG test, which is pregnancy-associated glycoproteins. And so as we go later and later, we can also use ultrasound. So as we move into the pregnancy, it's easier to study it. And I think most people on this podcast will be like, well, I'm sure Dr. Lucy is going to talk about this embryonic loss between 28 and 60. And of course, that's the one we know is a lot about because that's when we can very easily ultrasound cows and see it happening. But really, the bulk of embryonic loss that we deal with in dairy is not occurring here. It's occurring earlier. And that's kind of the new frontier, is understanding this embryonic loss that occurs earlier in pregnancy. If we go back now and look at Dr. Wiltbank's paper in which he summarized the losses, you can see that based on his paper, based on his summary of the data back in 2016, most people estimate that between 20 and 50% of cows that are inseminated lose the embryo within 1 week after breeding. In other words, you might start out with lots of embryos, but by the time they get through the oviduct, many of those embryos are gone and dead. We lose more embryos during this period of elongation and maternal recognition of pregnancy. And you can see a typical number of embryonic loss between 28 and 60 days. It's a real typical number, about 12%. So really, yeah, we lose a lot of embryos out here, but really we've already lost a lot of embryos before we get to this stage. And the important thing to try to understand is why. Why do we lose so many embryos early on? Now, in last few years, there's been a couple of really nice studies. This one out of New Zealand, in which they really tried hard to assess when embryos are dying, and they actually did this by flushing embryos out of cattle. And again, this is a study by Julie Berg. It's 1,500 grass-based cows in New Zealand. They estimated that the fertilization failure was about 16%. So again, about 85% of the embryos oocytes do get fertilized. You miss out on about that many. Then you lose another 15% between 2 and 7. In other words, by the time that embryo gets to the oviduct, you've lost 30% of the pregnancy. So if you flush a cow in New Zealand, at least, you're only going to get, after breeding 100 cows, you're only going to find 70 viable embryos. You lose some more after that out to day 15. And then in grass-based cows, you lose very few pregnancies after day 15. And so that was sort of interesting, that big difference that they don't, in grass-based cows, they're not losing a lot of pregnancies during the period that we think we lose a lot of pregnancies, later. So they lose most of their pregnancies early before maternal recognition of pregnancy. This is another study just published out of Ireland, and basically what they're also looking at is periods of embryonic loss. And in this study, they didn't flush cows during the early period. They're basing these percentages of loss based on sort of historical data in the literature. So they're saying up until the embryo enters, up until that morula stage, blastula stage, they're saying they lose about 27% of the pregnancies. And they say they lose another 15% of the pregnancies between the period of early embryonic development, elongation, and maternal recognition of pregnancy, and then another small percentage after that. So they say by the time they get out to 25 days of pregnancy, they've basically lost a half of their pregnancies. And again, they don't lose that many embryos afterwards. So clearly there's a lot of this embryonic loss that occurs early and not during the period that we typically think about embryonic loss. Now, I was curious about this concept that the grass-based cows in places like Ireland and New Zealand don't have as much embryonic loss between 28 and let's say 60 days. And this was a summary out of Santos et al. In 2004. He came to the same conclusion, that these grass-based cows just don't go through that same period of embryonic loss between 28 and 60 days that we see in our American cows. And that's an interesting question. I mean, the question is why? Why don't they lose embryos during that period? So the why of embryonic loss depends on when. So I think we understand about embryonic loss is you lose embryos throughout this early period before you get into placentation. And the question is why? Why do you lose embryos throughout this period? And so I'm going to just kind of talk a little bit about the whys. And so we talked about the first period of embryonic loss goes from fertilization to the development of the blastocyst. And so the question is the sire responsible for this? In other words, are there certain sires that better at fertilizing embryos than others? And that's a good question, right? You'd assume, well, there must be better sires than others. But there's not a lot of evidence for that. And what I would say about that is, assuming an appropriate timing of insemination with sires of proven fertility, there's just not a lot of evidence to support the idea that different sires have different rates of fertilization. So I guess what I'm trying to say is, regardless of whether or not you fertilize, regardless of the sire, you're going to get about 85% of those oocytes fertilized. And the other thing people think about is, well, does the sire contributing genetic defects to the embryo that's leading to failure for that embryonic development? Well, there are certainly genetic defects in embryos, and that's one of the reasons they don't develop fully, and that's probably a good thing. We don't want animals with genetic defects entering the herd, for example. And then there's this concept of lethal haplotypes, which are a form of genetic defect, which essentially basically makes it so the embryo won't develop. And we know there are lethal haplotypes in AI sires, but they only explain less than 1% of early embryonic mortality. So in general, we don't think it's lethal haplotypes or the sire contributing to that aspect of embryonic mortality. And then after that, it's kind of conflicting evidence from either Sophia Ortega's lab or this O'Callahan's lab, whether or not the sire is affecting the development of the embryo afterwards. And so it's just not very clear how important the sire itself is to this earliest phases of embryonic development. And when we think about things like sire conception rate, which is how we basically judge our sire's fertility, yeah, there's a range in sire conception rates, but I don't think we really know what part of the fertility that sire conception rate is explaining. What specific aspect of pregnancy is explained by sire conception rate? It might just be a lot of little things that lead to a higher fertility sire versus a low fertility sire. Now, on the oocyte side of the equation, there's a lot more evidence that the oocyte is compromised by various aspects of the postpartum cow. And this this concept was popularized by Dr. Jack Britt, who made the point that, the oocyte sits inside the cow for the entire postpartum period before it's ovulated, and that oocyte can be damaged by various things that happen to the cow. And of course, this toxic things, the toxic effects that can be felt by the oocytes, elevated BHBs and NEFAs, poor transitions, inflammation of the mammary gland or the uterus, and quite critically and clearly elevated body temperature. So when we think about the effects of heat stress on reproduction, it's pretty clear that the way that heat stress acts is by damaging the oocyte and making so it will not develop very well. Now, there's no question that a dirty uterus is not good for the development of the embryo. And the way we assess the health of the uterus is by a cytological examination. And so these are studies that you may see or hear about in the literature where they do something called a cytobrush exam. And a cytobrush is just a way to sample the cells that are inside the uterus. And if you see a lot of these neutrophils, which are immune cells in the uterus, if you see a lot of immune cells in the uterus, you know that the cow has a uterine infection. And quite clearly, based on lots and lots and lots of data, if the cow has a uterine infection, what you can see is that this is based on superovulation data, but what you can see is if the cow has a uterine infection, increasing amounts of neutrophils in the uterus, that the number of embryos that you can recover out of that cow declines severely. So any sort of uterine infection, it kind of makes sense, right? If the cow's got a uterine infection, you're going to have difficulties getting her pregnant. And that's kind of an obvious takeaway. So you need a clean uterus for that cow to get pregnant. And this is just simply additional data confirming that. This is out of Europe, but they basically did cytology at the time of AI, and you can see the drastic reduction in conception rate for any cow that has any of these immune cells in the uterus, indicative of an infection after breeding. So you definitely need a clean uterus to get cows pregnant. One of the things that's kind of been interesting, and I credit Pete Hansen for really bringing this forward, is that if we have so many embryos that are dead, like early after breeding, okay, if we have so many embryos that we lose, why can't we use embryo transfer to increase conception rates? Because you think about it, well, if you think about the New Zealand data, well, they said there was only 70% of the cows had good embryos in them a week after breeding. Why can't we just put a good embryo in those cows and increase conception rates? And that's certainly true in the heat stress example. In other words, there's lots of good example in heat stress cows that you can put an embryo in a heat stress cow and increase conception rate. But that's the only model system that we see, that putting an embryo increases conception rate. And the other thing I think that's really interesting, and this is from that paper out of Ireland, is when we put an embryo, even a fresh embryo or a frozen embryo, into a cow, we're still losing 30% of those embryos before maternal recognition of pregnancy. In other words, basically, even if we put a perfectly healthy embryo into that cow, we're losing a third of them before maternal recognition of pregnancy. And so even with embryo transfer, there's a tremendous amount of embryonic loss. Some of the embryonic loss, this is data published in JDS Communications, some of the embryonic loss is actually because if we do in vitro produced embryos, there's a lot of chromosomal damage associated with in vitro produced embryos. But even in this study where there was actually fresh embryos transferred, we're still losing a lot of embryos. And so I think what this kind of tells me is we don't really understand what a healthy embryo looks like under the microscope, just like we don't fully understand what a healthy spermatozoa looks like under a microscope. If you think about spermatozoa, yeah, we can do all the morphology you want, but in the end, you're gonna have to put it into a cow to see, get a sire conception rate, because that's the acid test is put into a cow. And I think embryologically, I think all embryos look the same, but I think there's probably something different about some of these embryos that confers whether or not they're able to get a cow pregnant. So I'm going to talk about the second period of embryonic loss, and that is between elongation and maternal recognition of pregnancy. And this is all about progesterone. So this period between like the blastocyst to maternal recognition of pregnancy is all about the hormone called progesterone. And I think we hear a lot about progesterone in dairy because people talk about, oh, dairy cows have low progesterone, therefore they have low progesterone, they don't get pregnant. And the reason progesterone is so important is because basically progesterone stimulates uterine function, and that additional uterine function supports a more rapid development from like the hatched blastocyst to the elongated embryo, which produces interferon tau. And this has all got kind of started way back when at Oklahoma State University. I'll give a shout out to my colleague Rod Geiser, and he's at the University of Missouri now. And basically what they found is that if they supplemented progesterone early after breeding and increased that progesterone relative to control, that they could stimulate the growth of the embryos. And this is kind of what it looked like in their study that they published. This is a control embryo, and this is an embryo that they took out of a cow that was supplemented with progesterone. Now, importantly, if you supplement with progesterone, the embryo develops more rapidly. If it develops more rapidly, it produces more interferon tau, shown here on this fluorograph, compared to the control embryo. And of course, interferon tau is critical for the whole story of maternal recognition of pregnancy, so that the cow stays pregnant. And so what's interesting about progesterone and supplementing progesterone after breeding to increase pregnancy is all the data that came out of Pat Lonergan's lab at UCD in Ireland with his colleague, Dr. Sanchez. And basically what's fascinating about the progesterone story is you can put together a tremendous story that demonstrates if I supplement for progesterone, I can increase the growth of the embryo, particularly to that filamentous stage, and they'll produce more interferon tau. But the strange part of the story is even when you do that, when you attempt to do that, you only marginally affect fertility. And so this is kind of curious. You can stimulate the embryo to develop and make more interferon tau. End of the day, you really see no benefit of supplementing progesterone to increase the fertility in cows. And so these are data, this one, Yanino, this is out of George Mann's group, published in Theriogenology. 84 experiments summarizing supplementing cows with CEDAR devices first week after AI, 3 to 7 days. And basically what you see is that they conclude no benefit of progesterone supplementation for cows with good fertility, no benefit to cows on zinc programs, maybe some benefit to cows in low fertility. So they're really low fertility cows, maybe you'll get a little benefit, but it's very small. Similarly, your other option here is to either treat them with GnRH or HCG after AI during the luteal phase. Same sort of thing. No difference between HCG and GnRH, no benefit to cows with good fertility, maybe some benefit to cows with low fertility. So that's been kind of an interesting story that's evolved, is even though we know these early embryos depend on this, they don't really, you just don't get a big lift by treating a cow with progesterone. You don't get a big lift in terms of fertility. Now, in the last section of my talk, I want to talk a little bit about placentation, and this is kind of what we work on in my lab. We work on sort of placentation, embryonic loss. And so in his summary, Dr. Wilbank discussed the risk factors for embryonic loss between day 28 and 60. And again, I want to reiterate that this is the embryonic loss we're looking at with ultrasound. It's usually 12 to 14%, somewhere in there. This is not the major period of embryonic loss that we really need to start focusing on. We need to really focus on this early period of embryonic loss. But Dr. Wilbank brings forward that failure to cycle is a risk factor for embryonic loss. Parity, first parity cows have more embryonic loss than multiparous cows. Farms differ for embryonic loss, that's very clear. Change in body condition, more body condition loss leads to more embryonic loss. And then uterine disease, which I want to talk about, but really the truth is any sort of disease, any sort of inflammatory disease leads to embryonic loss. This is particularly true of mastitis. Cases of mastitis around the time of breeding, it's been very clearly shown by several studies, typically lead to infertility and embryonic loss. So I'm going to talk about uterine disease today, and I'm going to talk about a few different mechanisms through which uterine disease leads to embryonic loss. So the reason we're super interested in uterine disease is because it's kind of interesting, right? The cow gets metritis 7 to 10 days postpartum, and then they usually get better. But if they get better and everything seems fine, then how come they don't get pregnant? Or why is this, consistently seen in the literature that sort of metritis leads to a reduction in fertility? Now, it's not complete infertility, but cows with metritis generally are harder to get pregnant. And so we think about lots of different things: residual infection, if they're still infected, morphological memory, inflammation, and cellular memory. And the first one I want to talk about, in general, cows that have had metritis, they generally, by the time you get out into the breeding period, they're generally not still infected. They usually can resolve that infection. So you're not dealing with, generally, not dealing with bacterial infections during the breeding period. You're dealing with other stuff in cows with metritis. The first thing you're dealing with to understand about metritis and how it potentially can affect early embryonic development and loss is this morphological memory. And basically what I mean by that is metritis damages the uterus, and that uterine damage is not resolved by the time the cow reaches the breeding period. And I want to focus, I'm not going to focus on the adenomyosis here. I'm just going to focus on scar tissue formation. And so this is an experiment. This is a paper we have in press in JDS Communications. And we're looking either healthy cows or metritis cows, either at 30 days postpartum, or particularly what I think we want to focus on is this later period, later postpartum during the breeding period. And what you see is cows with metritis, we're staining for basically collagen, which is scar tissue formation. Cows that have had metritis, there's a certain amount of collagen or scar tissue that's left over from that disease. And that's just kind of how it works. When you have disease and tissue injury, a lot of times the cells lay down scar tissue. And then scar tissue isn't the same as secretory tissue. So it's some of this embryonic loss is just probably because the associated with early postpartum metritis, is probably because the cow has just damaged her uterus and it just isn't as good as supporting the pregnancy. The other part of this thing that's important is even though these cows are no longer infected, the uterus still contains remnants of the previous infection. And this is we call this kind of residual inflammation. And what do I mean by that? Well, cows that have previously had an infection, the uterus sort of leaves behind these lymphocytic foci because it's a little bit concerned it's going to have another infection. And what the foci do is there's little clusters of immune cells that are prepared if the cow is going to get infected again. And think about these little clusters of immune cells. When we studied this in postpartum cows, and this experiment, we had postpartum cows, and we were breeding them and then slaughtering the cows and weighing the placentas from 28 to 42 days of pregnancy. When you see these little clusters of immune cells, we call those cows with high number versus low number, you can see an effect on the weight of the placenta. In other words, cows with a lot of these immune cells still present, inflammation still present, they had smaller placenta at both 35 and 42 days. And we think that's important. The other thing we notice if we look at inflammation, number of foci, or fibrosis versus number of foci, these two cows had embryonic loss. This cow, number 34, had a very small placenta. You can see that these cows that had embryonic loss, they had a lot of inflammation, a large number of foci, and a lot of fibrosis in the uterus. And so some of these things, is inflammation and fibrosis, they're probably associated with the inability of the cow to maintain that pregnancy. So we think what happens is as the embryo develops, basically it needs to communicate with the uterus. And if the uterus has still got foci there or elements of inflammation or scar tissue, it can't really do that effectively. And if it can't do that effectively, it'll develop for a period of time, but then stop developing. I guess the good news is when we study this, this is a experiment that we just did, and we just completed this work. Now, this was a entirely different group of cows. We're looking at the number of foci. We're looking at cows that were slaughtered about 80 days postpartum versus cows that were slaughtered about 165 days postpartum. You can see that at 80 days postpartum, the cows that had metritis, again, this was the first week postpartum, they had metritis. Even 80 days later, they're quite different from the healthy cows. But by the time you get out to 165 days later, a lot of those differences have gone away. So it's possible that time can heal some of these wounds for these cows and allow them to get pregnant. I'm going to skip forward through this, but the last thing I just want to mention about this is so there's when you have a cow with metritis, by the time you get out to the breeding period, there's still issues with inflammation. There's issues with scar tissue. All these things kind of affect how the uterus functions itself. And we definitely have data support this, and we just published this in Biology of Reproduction. And if you just read the title, metritis and uterine disease microbiome are associated with long-term changes in the endometrium of dairy cows. And what we mean by that is we can definitely measure an effect on how the uterus functions, and we can definitely measure that effect within the caruncular endometrium. And the reason that the caruncular endometrium is important is that's where the placenta forms. That's where the placenta of the fetus attaches to the placenta of the mother. And that's where we see these changes in gene expression. So we think that our hypothesis is that this disease is somehow affecting the way that placenta interacts and forms, and it does this well after the cow has already recovered from the metritis. So I'm not going to be able to go through that. So Pete Hansen in a perspective article, I really like this, was can early embryonic loss be reduced in lactating cows? And he published this paper in the Journal of Dairy Science 2023. And he said, yeah, we can reduce embryonic loss in lactating dairy cows by adopting best management practices for dairy reproduction. I've already talked about that. And developing new and innovative tools based on sound science. So the other part of this is he talked about, okay, are there different ways, if we do a lot of science, can we figure some of this stuff out? And I thought I would just leave you with this. While we wait for science, follow Dr. Fricke's rules for getting cows pregnant. And Paul and I are good friends, and we talk about all sorts of stuff. And we were in Ireland on sabbatical together, and we were at a conference that's over 10 years ago now. And Fricke went off this one. I just love it. How to get cows pregnant, you know, rule 1, put semen in the cow. And then rule 2, you know, if the cow's not pregnant, refer to rule 1. And that's really true, okay? Like, you got to get semens in the cow. And I'll just say cows with embryonic loss are not pregnant, so you should apply rule 2, which means referral to return to rule 1, get semen back into the cow. And so a lot of the times when I think about this question, and this is a paper I did forever ago when we were, I don't know, talking about PAG testing and stuff, is people would always argue, hey, I don't want to preg check too early because I'm going to be dealing with embryonic loss. You know, I don't want to check cows. I don't want to preg check cows at 28 days because I'm going to be dealing with a lot of embryonic loss, and I don't want to just have a cow preg check pregnant and then lose the embryo. And I would always argue, well, that's fine, but we don't really do pregnancy diagnosis to find pregnant cows. We do pregnancy diagnosis to find open cows because I can manage a cow that's not pregnant. You know, I can do something with a cow that's not pregnant. There's nothing I can really do to prevent embryonic loss at 28 days after breeding. But man, if I know that cow doesn't have an embryo in her, I can do something. And I think the latest data that shows that, you know, 60 to 70% of cows at 18 days after breeding probably don't have an embryo in them, that really makes you think. In other words, I've got most of my cows that don't have an embryo in them within 3 weeks that I can for sure know within 3 weeks after breeding. And is there anything I can do with those cows? So most guys, and this is according to Dr. Fricke's protocols, and I know there's a lot of different ways you can do resync, but you know, most guys might do something like a 32-day resync, put collars on the cows, do a pregnancy diagnosis, catch the cows that come back into heat naturally with the collars, do a pregnancy diagnosis, and those that don't, and anything that's open, just put them back on OpSync. And that's a really nice program. And what this does is it basically ensures that you don't violate rule 2. Basically, if the cow is not pregnant, you're going to put semen back into the cow. And this is just kind of gold standard how you do this kind of work. And the thing that's really interesting, and there's been some evolving in terms of pregnancy diagnosis recently, and that is our ability to do pregnancy diagnosis at 18 days after breeding. Could we do pregnancy diagnosis earlier than ultrasound and earlier than a PAG test? And the reason that question is really important is because if you can do a pregnancy diagnosis earlier, around 17 or 18 days after breeding, then if the cow is not pregnant, it's a real nice period to give prostaglandin time breeder so that you can get her re-inseminated within 21 days, right on that 21-day mark. And Jake Green and I did these experiments quite a while ago when he was a master's student with me, demonstrating that, yeah, if you could do a pregnancy diagnosis day 18, then you could resync those animals and get them rebred by day 21 and keep them on the same calendar as all the other cows in the herd. But the problem was that this day 18 pregnancy diagnosis, which is based on ISG15, which is basically measuring interferon tau, was really difficult to do. And then I saw this paper out of Milo's lab, which I really enjoyed. And what Milo and his large group demonstrated here was that there's a better way to measure this ISG15 compound. And basically, to remind everybody, the embryo produces interferon tau, which increases ISG15, and that's your measure of pregnancy diagnosis. When we were doing our work with Jake Green at the University of Missouri, we were measuring this in blood samples. But what Milo showed was that, well, if you just took a sample of the cervix, the vagina, you get a much stronger signal. You get a much stronger signal of the pregnancy, and that pregnancy can be detected as day 17 after breeding. So I'm really curious to see if whether these super early methods of pregnancy diagnosis can be maybe commercialized in some manner so that we can do a super early pregnancy diagnosis and basically resync all those cows that don't have, that do not have an embryo in them, that are absolutely not pregnant. So what it would work like this is if you could, based on some of these more sensitive early tests, if you check cows on day 18 and they were not pregnant, you could resync them and have them rebred on a weekly calendar. So they would fit right into your regular calendar. And then for the pregnant ones on day 18, well, you just leave them alone. You don't do anything. Keep the collars on them, recheck them, and just if they lose those embryos, rebreed them with the rest of the group. So it's kind of an interesting, twist to this, is whether or not we can basically, since now we know we've got so many cows that don't have embryos in them early, whether we can come in and just intercept those cows early on and get them turned around about 2-3 weeks faster than the rest of the non-pregnant group. So I don't know, I'm just kind of curious to see where that goes. That's my story. Appreciate everyone listening. For the latest in dairy research, listen to Dr. Lucy's Dairy Digressions. We do science right, and we talk about all sorts of stuff dairy. For those interested in H5N1, we have an H5N1 episode coming out with Diego Deal. We talk all things H5N1, and that should be coming out this week. Thank you very much for the opportunity to talk. Thank you.
All right. Thank you very much, Dr. Lucy, for that background information on embryonic development and then embryonic losses, and sharing some of the research that you and other people have done looking at this area. Sounds like there is plenty more for us to learn, but I think what you shared is really helpful for people today. So thank you. If you would like to view this webinar or any of the past Hoard Dairyman webinars, you can visit our archives at any time. Those are available on the hoards.com webpage, and just look at the webinars tab. And all of our previous webinars are located there, and this one will be available towards the end of this week. So if you want to watch it again or share it with somebody else, you will be able to do so in a couple of days. I would like to invite you to our upcoming webinars. The Hoard Dairyman webinars always take place on the second Monday of the month at noon Central Time. On March 10th, we'll have a presentation by Dr. Kirby Krogstad from The Ohio State University, and he'll be talking about low forage diets for dairy cattle, especially during this time of unique weather conditions where a lot of parts of the country are facing either drought conditions or maybe less than ideal growing conditions, and how dairy cattle diets may need to be adjusted to work with the feeds that are available. Then in April, we'll have a presentation by Jimena Laporta from the University of Wisconsin-Madison, and she will be here to talk about heat stress in dairy cattle and abatement strategies to minimize the impacts of that heat. Now we will tackle the questions that came in, and Dr. Lucy, we have quite a few, so I will just get started and we will move through these. So first question, if you want to move the slide ahead one, please. Okay. Thank you, and then we'll jump into these questions. Do cows with smaller embryos have greater loss of pregnancy?
Yeah, so I think they do. I think the Dr. Pursley has been doing some really nice, and Milo have been doing some really nice work on this. And what they do is they track that with pregnancy-associated glycoproteins, which is the pregnancy test you might use on blood or milk. And they definitely, those ones that come up later, that increase that later, are definitely at risk for loss. And we don't really understand I tell you, we don't understand the why of that. I mean, why do some come up later than others? But yeah, that's the concern. If they come up late, they're not developing at an appropriate rate, and they're going to lose those embryos. One of the things I forgot to mention, and perhaps some of the people on the webinar have an opinion on this, but I did ask Dr. Fricke as well as Dr. Polk, what do I do if I ultrasound a cow, let's say she's 32 days after breeding, and it's obviously a dead pregnancy. I don't see a heartbeat or the placental is detached. And both of them commented that you just continue on with the synchronization, the resync on her, and don't get your mind all wrapped up that, hey, this embryo's dead. I don't know if I should resync her or not. Just resync her, And the point of the matter is, if you take her off the resync program, your pregnancy, your conception rate on that cow is 0. If you keep her on the resync program, your conception rate will be something greater than 0, and it might actually be reasonably good. So that's the thing I forgot to tell. Thank you very much. Sorry about the digression there.
Thank you for answering that. What about an abnormal number of chromosomes? Can that be a cause for embryo loss?
Yeah, so that is, I tried to look that up right before. That's an excellent question, because I thought about that. And I tried to look that up, and I found, for example, in the IVF embryos, the in vitro produced embryos, yeah, you expect about 5% of those to have abnormal chromosomes. And I think there's been study in vivo, and yes, that is part of this loss. In other words, I think some of the ones that don't make it during this early embryonic development period are just wrong. They're not supposed to make it, but I couldn't find the data to say what percentage did have chromosome abnormalities. And there's not many studies on that, because again, just this issue where you have to really flush the embryos out to get that information.
Sure. At what day is tubular recognition of pregnancy?
Yeah, that's a good question. So the embryo starts to make the interferon tau very early, but we normally think about that period 16, 17, 18 days after breeding it starts, and that ends 24 days after breeding. So then the interferon tau is only present for a very short window of time. And its main job is to prevent the release of prostaglandin F2 alpha from the uterus. Now, the reason I said that you can resync cows then, okay, and I know that's sort of crazy and out there, but if you knew whether or not she was pregnant, that's what you've got at that point in the estrous cycle. If the cow is not pregnant, there's a corpus luteum there that's fully responsive to prostaglandin, and there's a second wave dominant follicle that's also present at that time of the cycle. So it's actually kind of a nice time to do a rapid resync type of thing. But the trick is, you need to know whether or not she's pregnant, and that's where this ISG15 test could tell you whether or not she's pregnant, and then just give her prostaglandin, generates 2 or 3 days later, and breed her. And that's going to depend on, I think the thing that's so fascinating about dairies is guys are so good at getting lots of pregnancies. So, would you bother? I don't know, but it's another option that new science is opening up. How's that sound?
Mm-hmm. Interesting. Thank you. This question, it's a little bit longer. It comes from a veterinarian. Mm-hmm. Can you say something about the current opinion or results when it comes to the topic of blood pH and embryonic mortality? In our practice, we are seeing more and more problems with cows' acid-base balance being thrown off, both in the transit phase and when they're fresh cows. This imbalance goes in both the acidic and alkaline directions. What do you think? How big of an influence might this have on embryonic development or loss?
Yeah, so that's an excellent question. Unfortunately, I'm really embarrassed. I have no good answer to comment on that. The direction I would probably take the answer if I was to think about that is we know that uterine health is super important to this whole whole process. And anything, and uterine health is largely dependent on sort of the function of the cow's immune system. I mean, that's where it's all about, you know, getting a healthy functional immune system, because those are the heavy lifters for restoring uterine function. And so if the acid-base balance is in some way disrupting how those immune cells circulate, how those immune cells communicate, how they fight an infection, then I would definitely say that would probably be the first thing I would think about. Because I think the thing that's so interesting about this story, and this is why we studied, and I probably said it too many times already, but all this stuff that happens in the transition period, everything that happens in the transition period, even though that transition period doesn't go on, you know, however long, it really has long-term implications to fertility of that cow. And I think it's because of this notion of sort of memory, that tissues kind of remember what happened, you know, it has implications. I wish I had a better answer. That's a great question. I wish I had a better answer.
I think you covered it well. And again, just highlighting how important the transition period is for repro, milk production, overall health, certainly important. Is there a difference between heifers and lactating cows in terms of loss of pregnancies at the various time periods of development?
Yeah, that's a good question. So we typically don't think about, and there was a little bit of data from Santos. I showed a graphic there. He was looking at pregnancy loss in a heifer. I call them heifers, you know, a yearling animal. And he was saying there wasn't much in those. And then according to Dr. Wiltbank's summary, he felt that the first calf heifer was at risk for more embryonic loss. The first calf heifer is at risk for metritis, okay, and retained placenta. And so some of that risk for embryonic loss probably traces back to the fact that a first calf heifer has more risk of uterine disease. And if they have more risk of uterine disease, they're gonna, that uterine disease is a risk factor for embryonic loss. I didn't do a good job bringing this up, but I want to say something about this whole uterine disease thing and embryonic loss and fertility. Animals get metritis or don't get metritis, but what we really need to think about is where that uterus is 1 month later, at 30 days postpartum. That's probably what's really, really important. And there are cows that can get a roaring case of metritis, roaring case of retained placenta, and by 30 days postpartum, you open them up and the uterus is perfect. There are other cows that seem perfectly healthy at 7 to 10 days postpartum, and you open them up at a month and their uterus is full of pus. And what, according to our studies, what really matters is what's going on in the uterus at 30 days postpartum. In other words, and now cows that have had metritis are more likely to have endometritis at 30 days postpartum. That's true. So you want to do everything you can to prevent metritis in the first place. But what we probably want to really understand is, how do those cows that get some metritis get better and other cows don't? That's the question, because it's pretty clear that if they can get better, they will be okay. And I think that's where a lot of the metritis literature kind of gets stuck, because we basically summarize cows into two groups, metritis or not metritis. But really, we need to know what those metritis cows did during that first month postpartum, because some of them just do fine with it. And that's true of retained placenta too, which seems crazy to me. But we just did a study in which we looked at that, and it was like half the cows with RPs, 30 days postpartum, they were fine, which is interesting. You'd think they'd still be kind of wrecked, but not true.
That is interesting. What is your opinion on the role of vitamin C, omega-3, or protected methionine supplements on embryo development and quality?
Yeah, so the vitamins to sort of stimulate immune function, and then the different fatty acids to sort of prevent the different fatty acid supplements to prevent to sort of control prostaglandin release, the methionine for the embryonic development. So the methionine, I know there's a lot of interest in that, and that has to do with just sort of embryonic genome activation. So the embryonic genome, in order to be activated, it has to be basically, there's a process involving the methylation of DNA, and that process has to go right in order for that embryo to kind of turn on its genome correctly. And to be truthful, in the human literature, most people will argue that when a woman loses an embryo, for example, a lot of that is chromosomal abnormalities, but a lot of it is the embryo just doesn't successfully turn on its genome. It has to start doing stuff with its DNA so that it can develop. I mean, it has to unpack it all and get it working. And that's where it kind of breaks down. The methionine is kind of designed to help that. The vitamin C, I think, is designed to sort of stimulate immune function. When anything you can do to stimulate immune function is good. And then obviously the omega-3 is designed to sort of control the inflammation. Anything you can do to control the inflammation is good. So those products are targeting the important elements, and it's not an area that I study. I haven't looked at the huge meta-analyses to show whether or not they benefit or don't benefit, but they're targeting the right areas. That concepts are correct. They're legitimate. I just haven't looked at the meta-analysis to try to understand the overall, your expectations for fertility on those things.
Thank you for sharing your thoughts. The questions are flying in. We'll address a few more of them and hopefully cover most people's questions. The next one is does a GnRH shot at AI breeding off a natural heat show much improvement in pregnancy and in maintaining that pregnancy?
Yeah, so that's a good question. So I think way back when with Jeff Stevenson at Kansas State, quite a lot of work on sort of GnRH treatment for repeat breeder kind of cows. And as I remember, there was a benefit for GnRH treatment for repeat breeder cows, at breeding, if you're doing a natural service, GnRH treatment. And I think that comes down to the fact that some of those cows are delayed ovulators, and so you try to just prevent that with that. I'm most familiar, to tell you the truth, with either GnRH treatment or HCG treatment after breeding, like a week or 14 days or somewhere out there. And again, the large meta-analysis says those will help, but only in problem cows, low fertility cows. And so, and I think the reason they probably help in low fertility cows is there's probably a population of cows out there that do have low progesterone, and additional progesterone will help. And but I think where we kind of get stuck on that is what cows do you treat and what cows don't you treat, And if you had a definitive way to do a very, very rapid test on a cow to say that, okay, this cow is below the norm for progesterone, yeah, you'd probably hit her with HCG or something to get her up a little bit, if you wanted to, if that was what you wanted to do in your herd. But there's just no easy way to do that, So there's just sort of like, you look at her and you're not going to blanket treat the whole herd with, for example, like HCG is a good one, a certain number of days after breeding. You're not going to blanket treat the whole herd, because I think the data would say most of the herd don't need it. You know what I mean? So you're treating a lot of cows that don't need it. But again, the meta-analysis, which were quite large, said that, yeah, those treatments do help, but only for problem low fertility cows. But that's a good question.
All right. Could embryo transfer help us with embryonic loss during the heat stress season?
Yeah, for sure, definitely. That is the one. That's iron. That's solid. Iron solid. Okay, so no question. Pete Hansen's done a lot of work on that. Issue is that the heat stress just hammers the oocyte and hammers the early embryo, hammers it, and so you'll go from a 10% conception rate in the summer to 20, 25, maybe even 30 if you do an embryo transfer. You'll never get back to the wintertime conception rates, but you'll move the needle. Now the problem with it is not the problem, but the source of embryo and inexpensive embryos to really make it sort of I don't know. I don't know if anybody does a commercial. I'm not smart enough to know that, but it's the price of embryos. That's the thing. Are you going to really spend all that money on embryos if and then of course, there are commercial operations that provide in vitro produced embryos for this purpose, frozen in vitro produced embryos for this purpose. And I think the thing to think about, I skipped over this slide, but if you look at the slides, there's some groups working on producing bovine embryos from stem cells, and you're like, oh my God, Dr. Lucy, what are you talking about? Well, you can make stem cells, you can make lots of different types of cells with stem cells, okay? And so, you can make bovine embryos with stem cells. And so what that would do, if the technology ever got there, that would if you could produce a viable embryo from stem cells. Now we're not doing flushing, we're not doing in vitro produce, we're making embryos from stem cells. Then you could produce a lot of embryos for transfer. And those embryos might be the price point that might make it worthwhile, maybe, to put them in heat stress cows. And then the other thing is that we just don't do enough embryo transfer to understand whether or not some of these other issues could be solved with it, and but maybe that's the future, but that's got a little ways to go yet.
Okay, one more question for your consideration. Have you noticed, or do you know if there are any appreciable differences in embryo or fetus retention when blood or milk is utilized to diagnose pregnancy versus ultrasound when both are assessed at later than 30 days since the last heat?
Yeah, so not that I know of. The real, I think we all, I've been in this business for a long time. There's no question. The nice thing about ultrasound is you get a diagnosis of embryonic mortality at cow side. That's really helpful because you can resync those cows, and you don't get that with blood or milk. I mean, you just don't get that. And so I'm not sure, the blood and the milk tests, they're really related tests, but their sensitivities have been tweaked. So I don't think there's any major differences between, if I was to guess, I would guess the blood test is more sensitive to the milk test. And when I see more sensitive, I mean, you can probably pick up cows earlier after breeding with the blood test. And the reason I say that is because the PAG concentrations in blood are much higher than the PAG concentrations in milk. Okay, so you're going to be able to see a cow pregnant much earlier with a PAG test than a milk test. The milk test is really designed for convenience, but neither of those tests can tell you whether or not that cow is undergoing embryonic loss. Now, if you had a blood test or milk test that would tell you that cow is either pregnant or not pregnant, or undergoing embryonic loss or not, that'd be handy. But again, the situation with the blood test is, or excuse me, the blood or the milk test is there is a certain amount of variation in the strength of that signal. In other words, it's not perfect one-to-one. I mean, you got perfectly healthy pregnancies that produce, 10 nanograms per mil of PAGs. There's a range in normal. I'm just trying to say this. For these PAG tests, there's a normal range. And, they got to be pretty low before, in my opinion, they got to be pretty low before you'd be like concerned, like kind of in that recheck level, that type of thing.
All right. Well, thank you so much. And thank you to our audience. You submitted a lot of great questions. Dr. Lucy's contact information, his email address is in the presentation slide. So if you have some follow-up questions, feel free to message him, and I'm sure he would address those as he is able. But once again, thank you to our speaker, Dr. Matt Lucy from the University of Missouri. Really thank you for your great presentation and then answering all these questions. That extra insight really helps get the message across and provides additional information.
Yeah, I really enjoyed it. So thank you very much. Really fun to give a talk here. Thank you very much.
Thank you. I also want to thank all of our listeners for making time to tune in to today's program. I hope that you heard something that can be put to use on your farm or on the farms that you work with. Until next time, goodbye from the team here at Hoard's Dairyman.